Okay, class. So this is lecture 3, and then this is the first part of that. So we'll just call it lesson 1. This lecture is going to focus on RNA and RNA processing. So we talked about DNA replication, and now we're going to talk about transcription. So how does DNA become RNA? What's the purpose of that? And then we'll lead that into the next lecture, lecture 4, which is translation. So transcription now, and then translation in your future. So DNA directs this synthesis of proteins ultimately, and we talked about the central dogma. So I'm probably going to ask you on the test. Central dogma is DNA is turned into RNA, and RNA is turned into protein. And I know we've already gone over this, but I'm going to go over it again because it's super important. So this is called transcription, and this is called translation. And in transcription, so the DNA is in the nucleus of eukaryotes, and the RNA has to go into the cytoplasm. It's a mediator to get to the ribosomes. So the ribosomes are what does translation. And then transcription is done by an RNA polymerase. We already talked about DNA polymerases, and now we're going to talk about RNA polymerase. So this is a DNA dependent RNA polymerase because it needs DNA to make RNA. Hopefully that makes sense. All right. So we've talked about the sequence of DNA forms genes, and that it makes up our genotype. So, whatever letters it is that make your proteins fold into a shape that makes a brown pigment, it gets deposited in your eyes, makes your eyes brown. And the default pathway for eye is blue. Same way with Mendel. Remember we talked about in Mendel's world, the purple pigment is DNA codes for an RNA that's made into a protein that folds into a shape that transmits purple wavelengths to your eye, and so that you see the flowers purple. By default, if that is broken in any way, then the default pathway would be white flowers. And so that's our genotype. So we're talking about, big P, little p. These represent chromosomes, homologous chromosomes, not sister chromosomes. So this would have an allele at a locus. We already talked about this. And this would have an allele at a locus, and these two alleles would code for flower color, right, which is the trait. And that's the phenotype, what you actually see. You can't see inside the cell, so you can't see the genes. But this one, if it's big P, codes for an RNA that makes that is converted into a protein that makes a purple pigment, right? And this one, little p, is broken. It could be broken at many places, but for whatever reason, it's not read from DNA to RNA. It's not processed into a proper RNA. It doesn't fold. There's lots of stuff that can go down that would cause this to fail. And then again, by default, it's white because purple is broken. So everybody knows genotype and phenotype. And that proteins perform a specific function. It could be enzymes, like proteases or whatever. It could be stability, like the cytoskeletal proteins, like actin and tubulin. It could be a hormone, like insulin is a protein. So whatever it is, that produces the phenotype, which is in this case, this would be purple, right? Because it's a heterozygote. All right, so transcription and translation are the processes linking DNA to proteins. We just talked about the central dogma. DNA is transcribed into RNA, and RNA is translated to the protein. I think you guys got all that. All right. So we've already talked a little bit about this. What's the difference between RNA and DNA? I think you have a good background on this. RNA is usually single-stranded molecule. Sometimes it can be double-stranded. That's usually viruses. And you're going to do a little exercise on your problem set for that. So it can bend into complex three-dimensional shapes. So think about RNA like this, right? So we have to label the ends. RNA is going to be 5 prime to 3 prime. It's always going to get made this way. That means the DNA has to look like this. So this is the DNA, and it's converted into the RNA. And it would be, so if this was like this, it would be read this direction. An A would be converted, not a T. There's no T's here, but that would be a U. And then G would be a C, and so on and so forth. So that would just be like translation until you get to the end. So some things that you need to keep in mind here is that DNA, we have a coding strand and a non-coding strand. So the coding strand is the strand that has the same code besides U's in the RNA. So in this case, remember DNA would have a complementary strand, and that complementary strand of DNA would be T and C and so on. So this top strand of DNA right here, this one, is the non-coding, and this one is the coding strand. All right. So because the code, right, let's just say we go on with this G and A, whatever. This is, so this would be a C and this would be a T, and then this and would be a C and a U. I don't know, my computer's lagging for some reason. C and a U. So this code right here is the same as this code, except we're replacing the T's. And that's why it's called the coding strand. The non-coding strand is the one that isn't the same as the code, right? So in this case, the strand that's red is the complement. The non-coding strand is the complement to the RNA. The coding strand is the exact same as the RNA. Same orientation too, 5 to 3. So most of the time, whenever you're looking at DNA sequences, like, let's see. We'll just look at something like, I don't know, Bordetella pertussis, sorry, Bordetella pertussis. Um, I want to see its whole DNA. So there's tons of information in here. I just want to know the nucleotide sequence. I'm just going to use this as an example. Okay, so this is the whole genome. This gives you the source organism. It's kingdom, phylum, class, order, family, genus, and species. This is the scientist that published this article and sequenced the whole genome, and then this sort of gives you the reference of it. So we just want to get the sequence. So we're going to click on Fast A, and that will give us the whole sequence. So this sequence right here is only a single strand, not both strands. And because this sequence is identical to the RNA, it's written in the 3' to 5' direction. And so this is the coding strand, not the non-coding strand. And all DNA that's published is generally the coding strand. And the reason is it's a lot easier for me as a scientist to go, okay, what's the RNA look like? Okay, it's U A, oh sorry, A U G G A U U U U C C C C C G C G A A U U U G A T. Oh sorry, U yeah U, because it's identical to the sequence that's published. I don't have to go, okay, A pairs with T and G pairs with C, and there's no T's in RNA. So the coding strand is always the one that's published. The non-coding strand isn't published. Um, all right. And RNA is more like a string. Okay, so RNA, like DNA, right, is more like a string than a piece of steel. So RNA can fold. It can make what we call stem loop structures. And if you've ever seen like a tRNA, you'll you can notice that um, it has these stem loop structures. So we'll talk about it. So RNA can make complex three dimensional shapes, more complex than DNA can. So like a protein, and unlike DNA, RNA can uh, catalyze or speed up reactions. And those kinds of RNAs are called ribozymes. So one of these that we're going to talk about is in a spliceosome, but they're RNA enzymes. So these RNA enzymes. Let me pause it. So we talked about life. We talked about the evolution of life. We talked about that all living things use DNA as their genetic material. So we also talked about DNA is unique because it codes for its own replication. So then that's kind of the chicken and the egg thing, right? So what came first, the DNA that codes for the protein, or the protein that ended up coding for the DNA? And there's other problems with this. DNA can't catalyze reactions. Proteins can't really carry information. So they look for an intermediary to explain where life evolved from. And the conclusion is that life probably started from an RNA molecule similar to ribozymes. But there literally is no evidence for any of the that surviving. There are some things, but not living organisms, that can use ribozymes as both their genetic material and the catalytic material like proteins. All right. Okay. So we have transcription. Transcription is converting DNA into RNA, like we talked about. The enzyme that does that is an RNA polymerase. We already talked about it. This RNA polymerase can only copy single-stranded RNA. So it takes DNA and takes a single strand of DNA and makes a single strand of RNA. Translation is converting RNA into proteins, and then that's done by ribosomes that are found in the cytoplasm C. In bacteria, transcription is coupled with translation. So because there's not a nucleus, right? So they all occurs in the same place, and that means that bacteria are faster at producing proteins than we are. The DNA and the ribosomes in prokaryotes is located spatially in the same area, whereas here the DNA is located in the nucleus. It has to be processed before it leaves, and then it's exported out of the nuclear pore into the cytoplasm C, where it is translated into proteins. I'm just going to show this. And that causes a delay time in transcription and translation. Okay, what's a gene? We talked about genes. Genes are actually made up of pieces of DNA. So if we looked at a chromosome, like let's say your chromosome number one, you would, there's about 20,000 genes. So there's roughly 1,000 genes per chromosome. Let's just give or take. So there's all kinds of genes along here. And if we were to zoom into one of these pieces of DNA, there would be, we would have a 3' end and a 5' end, and then it's anti-parallel. So we would have the other strand. And so there would be letters, and A, G's, and C's, and T's. And on the complementary strand, A pairs with T, the C, G, A, T. I think that's a G I meant to be a G. Oh, I just butchered that. So that would be a C, and a C, and a G, and an A, and a T, and I think that's a G. So that would be a C. So these are our two strands of DNA, right? So what constitutes a gene itself? So a gene is something that codes for either a protein or a functional RNA, which we covered already, like tRNA or snRNA or miRNA or ribosomal RNA, right? So it could code for any of this stuff. All genes have three parts. They have a promoter. A promoter is the region. So let's zoom in on this. And let's just say we have this. And we're going to say this strand is the coding strand. So this is the strand. We're just going to make the upper strand the one. And I'll just put the other one down here just for reference. So this is one we're going to focus on. So the RNA polymerase is going to open this up. And in this area, we're going to have a promoter. So the promoter has a specific sequence in there, and it recruits what we call transcription factors. Makes sense, right? Because we're doing transcription to certain sequences in here. So you might have an estrogen response element, and that's only going to bind estrogen. So that helps turn on estrogen genes if estrogen's present in the cell, or testosterone genes, or whatever. So promoters have different characteristics. They almost always, and especially they almost always have like uh, a TATA binding protein product. So the sequence is TATA, and that is upstream of what we call the start site. So this is the start site, and after the start site, um, we have a terminator. That's a sequence that forms a stem loop that causes the polymerase to fall off. So we have the promoter. This area between the start site of the promoter and the terminator is called the coding region. And it is the area that codes for the actual protein. And then the terminator, like we said, is a sequence of DNA that causes the end of transcription. So basically, what happens, and I think I go into this in a future slide, but let's just say we have TATA binding protein. This is a very simple view. So TATA binding protein is a transcription factor. It kind of like the people that flag down the airplanes at the airport. It recruits the RNA polymerase. I'm just going to draw it as an egg, right? And it tells it where to line up to start. So any of the sequence in here, let's say that this is, A, T, T, A, T, A, G, C. Any sequence back here is not going to be made into RNA. And remember, RNA has to be made in a 5' to 3' direction. So it's going to start right here for transcription, whatever's beyond that is what is going to be made into an RNA. Nothing in front of it. So we call this nucleotide -1. We call this nucleotide +1 because it's part of the coding region. Anything that with a minus number is in the promoter or before the promoter. Anything with a plus number is going to be after the start site of the promoter. So let's say that this sequence is, I don't know, A, G, C, T, A, C, G, whatever. Then that is going to be converted into RNA on the mRNA. And that's going to be, so A pairs with T, but not in RNA. We have U, G pairs with C, C pairs with G, A pairs with T. Again, we need a U, G, C. This would be much easier if I had the coding strand, because the coding strand would be, and I'll show you, this area would be a T, right? A pairs with T, G pairs with C, C pairs with G, T pairs with A, T, G, and then C. So this is the, even though this says the coding region, this strand of DNA is the non-coding strand of DNA. This is the coding strand of DNA that's not being made into RNA at all. It's just the other strand of DNA. But you can see how the sequence is exactly the same. All I have to do is replace the T's with U's. And so you can see why writing the coding strand is much easier to translate than it is to go, what pairs with A, what pairs with G, blah, blah, blah. I can just go, this is U, C, G, A, U, G, C. And it's the same as the RNA. All right, so we just keep going until the terminator is hit by the polymerase. It knocks it off. So our egg falls off. This is our polymerase, RNA polymerase. It falls off because the terminator, it's kind of like derailing a train. So imagine you just threw a railroad tie on a train track and it knocks the train off. So that's what's going on. I don't need to get into too much specifics here. You'd say that for a molecular genetics class. Anyway, so that it gets derailed, and then the RNA is going to get processed. So that's what goes on in the promoter, the coding region, and the terminator. And hopefully that makes sense. Once all this, let's say this sequence is G, C, or whatever, then this would be C, G. I'm just going to use 3 dots here. We'll just use 3 dots here. So we just keep going until we get to the end. The terminator, that gets knocked off, so there's no more RNA being made. So this is the mRNA primary transcript. All right, so here again, these are different terms. So we have the coding strand, which I told you about, that matches the RNA. And then we have the non-coding strand, which is the complement to the RNA. And then, scientists love that. It's like things are society thinks they're important. They have lots of words for, like, think about all the different words for money. So same thing here. We use a template or non-template. Okay, so the template strand is the one that's actually being transcribed. So in this case, template is the same as the one that's being, so template is the one that's being transcribed. So template is also non-coding. They mean the same thing. Those mean the exact same thing. This is non-template means coding, right? Because the coding strand isn't being acted as the template. So when you're doing this out, I'm going to ask you on your problem sets to tell me what's the template, what's the non-template, what's the coding, what's the non-coding. Make sure you guys know the names of this. This is how geneticists talk, so I didn't make this up. They did. So you just need to learn it because it comes up more often than not. So here we have the non-template strand and the template strand. Remember, the template strand is the one that's going to be the template. So G pairs with C and so on. The non-template strand is the one that's not used as the template. And remember, we talked about, so template strand I said is the same as the non-coding, right? Because this is not the same code as this, right? This right here, the non-template strand, is the coding strand because the coding strand is identical to the RNA, except T's are converted into U's, right? So the coding strand is the one that's always written out, if you see it in a paper or whatever. All right. So just make sure you guys keep all that stuff straight. RNA polymerase makes a mRNA strand. It's complementary to the template strand that we talked about. Template strand is always 3' to 5' because no polymerase, not even RNA polymerase has to go in the 5' to 3' direction. So it has to go in this direction, and that means the template has to be 3 to 5. If it was going to use this as the template, the polymerase would have to go backwards. It would have to go 5 to 3 because it needs that OH group we talked about. This is just showing you, I think this kind of lines up, but so here's the initiation site, right? Everything to the right of that is +1, and everything to the left of that is -1. So when we're talking about minus numbers, this is just used to identify this is the +1 right here, right? Right after the initiation stripe. We say this direction is downstream, just like a river. This direction is upstream, just like a river. And that's because the template strand is going to be read in this way, which is if you threw a stick in a river, it would go this direction, right? Downstream, not upstream. So, sequence of the non-template strand is identical to the mRNA, which we talked about. Nucleotide positions downstream are indicated with positive, upstream is negative. This is just to kind of tell you where mutation is. So, if I'm talking to another geneticist and I'm like, oh, well, there's a mutation at -11, they know it's this one, right? Well, it's actually this one because this is probably -10. What's this one? If I'm saying there's a mutation in the RNA at +11, so this is 10. It should be 1. This is 10, so that would be 11. Right? So we're talking about the same thing. And when we write out mutations, we might say arginine is converted to lysine at nucleotide, I don't know, 231 or whatever. So then you know where the mutation is and you can go look for it. Like this is really handy if you're trying, so you don't have to go and you can look through a piece of DNA and figure out exactly where you're talking about where that mutation may occur. All right. So here's the deal with transcription. I kind of drew this out, but this is much better artwork than my artwork. So we have the promoter, right? Remember the promoter has a start point right there. And so everything in this direction is going to be minus numbers. Everything in this direction is going to be plus numbers. This is the RNA transcript that's being made. Remember, it's 5' to 3'. No polymerase can do it any other way. So the RNA polymerase comes on here from initiating of transcription factors. That's initiation. We'll talk about that. Elongation is very easy if it sees, it's reading off the 3 to 5. So if it sees an A in the DNA, it's going to put a U in the RNA, and so on and so forth. And then termination of transcription occurs from a different area. So we talked about the stem loop terminator that terminates or knocks off the RNA polymerase. So there's three parts to transcription: initiation, elongation, termination. If we look at promoters, right? So remember this is the +1 site, so this is the site first letter that's going to be turned into a RNA. This is everything that's not going to be turned into an RNA, so this is part of the promoter. And then if we look at roughly around the -10, we have what we call the -10 box. It's similar to the Tata box in eukaryotes, similar sequence. So TA. And then there's a highly conserved region that's called the -35 box. You can see it here. And again, we're going to look at the difference between prokaryotes and eukaryotes. That's super important, I think, because we have all of these prokaryotes that are antibiotic resistant. And in order to figure out how to attack them, we need to understand their genetics. So in prokaryotes, right, like E. coli, Escherichia coli is a prokaryote. It's a bacteria, lives in your colon, named after Escherichia, who is a doctor who discovered it in people's colons. So these are the highly conserved regions. These other regions in between here are free to mutate, and they mutate quite a bit, but these are highly conserved. So why are they highly conserved? Well, they're highly conserved because there's a sigma subunit that's part of this whole initiation complex. Remember we talked about transcription factors? In prokaryotes, we call it an initiation complex. And so there are 5 subunits that make a prokaryotic initiation complex. There's only one type of RNA polymerase found in prokaryotes. So just so you know that. So there's 2 alpha subunits found here and here, and then 1 beta here, 1 beta prime here, and then 1 sigma subunit. So this whole thing is the transcriptional initiation complex. The sigma factor, it's a subunit of the RNA polymerase. It binds specifically to this -10 and -35 sites. Kind of like, so if we have the sequence here, it kind of bends like this. It's kind of shaped like a U. And so this is the -35. This is the sigma 70. And this, I'm sure that's a Fedberg unit, so it doesn't have any relevance. It's just a logarithmic measure of mass. And then this is a -10. So that sigma 70 only touches and recognizes a few sequences around the -10 region in the promoter and around the -35 region in the promoter. And because it's not interacting with anything in between, it doesn't really care what that DNA sequence is at all. So once all of the factors are in place, and if you had mutations in the -10 and -35, this wouldn't be in place, so you would never get transcription. So once all these factors are in place, the polymerase begins making mRNA. The sigma factor is released. And so you can see this is the mRNA right here. That's 5 prime. And then, of course, I might ask you, okay, well, this is 5 prime. What's this? This is 3 prime, right? And so the end of this would be 5 prime. And I might ask you, okay, is this the coding or non-coding region or strand? And the answer is it is the non-coding strand because it's complementary. The coding strand is this strand here. What's the template strand? Well, this is the template strand here. What's the non-template strand? This strand here. All right, so this is just showing you, here's the RNA polymerase, here's the sigma factor that's attached to it, here's the DNA. This is, everything in green is the promoter, everything in pink is the transcribed portion. And you can see it starts right here. That's the transcription initiation nucleotide. This is the -35 and the -10, and you can see the sigma factor actually binds only to those regions of the DNA. It recognizes the DNA sequence and binds to it. That's why those DNA sequences are important. If you guys have any questions, send me an email. It's not that big of a deal. You'll get some practice on this with the problem sets too. And then, so after it's added in, remember it's starting here. So once it's started adding in, and the polymerase only opens small regions at a time. So around 30 nucleotides as it adds in the complementary nucleotides into there. So that's when the sigma factor is released, and then that's initiation, and then this is elongation. Stop doing this. Elongation. All right. So this is just another artist rendition of elongation. Essentially, we have the same area, 10 to 30 nucleotides at a time. And if it sees a T in the DNA template, it puts in an A. If it sees an A, it puts in a U, a G, it puts in a C, and so on and so forth. So this is elongation. It's not super exciting. Um, you can actually practice this if you want, I think, in DNA Interactive. So if you go to DNA code, here it is. It's being super slow. I'm gonna pause. I don't know, that's weird. It worked yesterday. But anyway, apparently it's not working, but you could pretend like you're a polymerase and you can add in the nucleotides. Uh, just like if you were an RNA polymerase. But apparently that doesn't work now, so you can't do that. But you can pretend if you want. Yeah. All right, so this is just showing you that um, genes are like a highway. So you can have multiple cars on the highway at the same time. You don't have to wait for a car to get off the highway before you can enter it. And it's the same thing here. So lots of RNA polymerases are going to be on the same DNA, the same gene at the same time. So let's say here we have our promoter, sorry, and our coding region and our terminator. So once one RNA polymerase gets on here and starts making RNA, it's out of the way. So let's say it's moved down here. Well, another one can jump on here, and it can also make RNA. So you have lots of RNA. So what's the direction of transcription, do you think? It's this way, right? Because it's getting longer on this end as opposed to this end where it's shorter. Same thing here. Direction of transcription is this way because it's getting shorter. It's short here and getting longer as it goes. Same thing here and so on. Okay, termination. It's a specialized termination sequence. It's a stem loop sequence. So it's an inverted repeat. So if you look at this, you'd see AAA, these pair with the T's on this end. This C pairs with this G, and so on, like that. So that forces it to become a stem loop structure. Um, and then this is the mRNA that's being created in transcription. It also has stem loop. DNA has a hard time making stem loops, but RNA doesn't. Remember, it can fold. And so that signals termination for the RNA polymerase at that termination sequence. So mRNA folds into a hairpin, like we said, and then that signals termination. Again, you can see it here. So this is U's, not us. So once this string of U's has been transcribed, that signals the RNA polymerase to fall off, and they know this because they've done mutation studies on it. So anyway, this is the hairpin loop that signals the RNA to be released and fall off, and that's how termination occurs. So we said there's only there's only one kind of RNA polymerase in prokaryotes. In eukaryotes, there's three. There's polymerase one. It's what transcribes ribosomal RNA, so rRNA. We already talked about it. Polymerase 2, that transcribes all of the messenger RNA, which we also talked about. And then polymerase 3 transcribes other small functional RNAs like the miRNA, the snRNA, and all those other ones that we talked about in the last lecture chapter when we talked about RNA. Okay. So we're talking about eukaryotic promoters right now. So promoters in eukaryotes generally have what we call a Tata box. This is the consensus sequence of it. Let me try a different color. This is the consensus sequence of it. Make this thicker. So it's usually TA, TA, A or T, A and A or T. Um, and I put this in here just so that you could see the variation in different genes in their promoter. So we're only looking at the promoter, right? We have the promoter, coding region, and terminator. So promoter, coding region, CDS coding sequence, and then the terminator is over here. So we're just talking about the promoter, right? Whatever's upstream, negative numbers, and you can see we number these negatives. So the Tata box is usually found between minus 20 and minus 35 in eukaryotes. In prokaryotes, remember we have the minus 10 and the minus 35 box, and that's where the sigma factor attaches. There's octamer motifs, so usually those are found in B cells. B cells are the cells from your bone marrow and your immune system that produce antibodies. And then we have the cat box. So this is fine, minus 70 through minus 200. This is the cat box minus 70 to minus 200 because it has the CAT, and scientists are trying to have a sense of humor. And then the GC box, so it has G's and C's in it, and that's usually minus 70 to minus 200. So, some of these things you need to be expressed all the time, and some of these things don't need to be expressed very often at all. So if we look at beta hemoglobin, that is not generally needed unless you're making blood cells. So it's not heavily expressed. And then here's the Tata box here. They all have a Tata box except this. SV, it stands for simian virus. Viral promote. So if you think about a virus, when it gets in a cell, what does it want to do? And the answer is it wants to get in, make as many viruses as it can, and get out. And so those are promoters that are usually always on. They're screaming on. And you can think of a promoter as kind of a dimming switch, on a light switch. So you could put it full on or off or anywhere in between. That's the function of the promoter. It promotes the expression of that gene, which simply means making it into RNA. And then we have kinases. If you don't know this should be a refresher, but a kinase is any enzyme, ASE, that adds a phosphate. So it adds a phosphate, it's a kinase. This adds a phosphate to thymidine, and so it's called a thymidine kinase. And so it has a GC box, it has a cat box, it has another GC box here, an octamer here, and so on. And then histones H2B, you should know that because we talked about that in the previous lecture. So this is one of the 8 subunits, right? There's 2 of them. This is one of the 8 subunits of a histone octamer. Histone octamer. Sweet. All right, so when we do genetic engineering, and this is how we can find out if your food is genetically modified. So when you design a gene, the gene has to have a promoter, right? It has to have a coding region and it has to have a terminator. Because you only want a specific amount of RNA made. So here's our DNA, right? Our gene. We want to put this, let's say this is salmon. Let's make it easier. Let's say that this is, okay, let's just say it's salmon DNA. And we're going to put it in strawberries. So this is going to code for a protein that resists freeze. Okay, so there's some things that we want to do this. So we're going to take the coding region for the salmon, right? Because we want that RNA to be made. But we're going to take a different promoter because in a salmon, this is probably not always on. What promoter is always on? A virus, right? So there would be some promoter that, well, let's just say one of the well-known promoters is a cauliflower mosaic virus promoter. So would you find a cauliflower mosaic virus in a salmon? Probably not. Would you find a cauliflower mosaic virus in a strawberry? No, because it's only in cauliflower. So we use this promoter quite often in biotech to make lots of the product because we want our strawberries to be really resistant to frost. And then the FDA is going to require us to make a really good, strong terminator because if the terminator doesn't work, it's going to make a different mRNA, right? It might be longer, it could be really long, and that might fold into a protein that kills everyone. So you don't want to have your gene like, people eat strawberries and they die. And they're like, oh, genetically modified foods, boo. So we have to have a strong terminator, and the terminator that's generally used is from a bacteria called Agrobacterium tumefaciens. I know this handwriting is terrible because my pad is like, I don't know what's going on with it. So Agrobacterium tumefaciens, I apologize. Um, that is going to be the terminator that's used because it's the strongest terminator we know. So it would be like, okay, there's some terminators that are like putting a penny on a railroad track. That's probably not going to knock the train off. But if you put a whole bunch of steel bars on it, something you was guaranteed to knock that train off the track, uh, that's the terminator that we're going to use when we try to introduce one gene into another organism. Because the FDA is not a fan of maybe it'll stop here and maybe it won't. Because we don't know what kind of genes it's going to make then. Maybe they're genes that kill you. So, or protein products that kill you. So they're not going to allow that. So when you're looking at this stuff, if I go to the store and it says non-GMO, the first thing I'm looking for is the DNA sequence that matches a cauliflower mosaic virus. And if I find that, 100% that's genetically modified, unless I'm looking at cauliflower and maybe that cauliflower has mosaic virus. I should be able to look at it and tell. And then again, if I'm looking at a salmon gene that's put in a strawberry, I certainly wouldn't find a bacterial terminator sitting in there. So I would look for the DNA sequence for this terminator that's specific for Agrobacterium tumefaciens that would stop this gene from being transcribed. Okay, so a little more about the promoter. Again, this is eukaryotes. So this is the Tata box. This is the Tata binding protein, eukaryotic promoter. So there's a protein that recognizes this DNA sequence. It's called the Tata binding protein, or TBP. Uh, it finds and binds to this promoter sequence. That's the representation of this. So this is the transcription factor. This is Tata binding protein, this shaded circle here. That in turn recruits other transcription factors. There's lots and lots of transcription factors. In fact, um, some promoters can have hundreds of transcription factors before everything's in place to actually start transcription. But these are the core ones. These are the ones that are found in almost all the transcribed genes. Here's another one there. Here's another one here. And so here's our start site in the DNA. So everything up here is the promoter, and that's a minus value. Everything to the right is a plus value. And then we remember it has to go 5 to 3, so it has to transcribe this strand. So what is this strand, coding or non-coding? The answer is non-coding because it's the template. Non-coding or template strand. And so by contrast, this would be the coding and non-template strand. The whole thing taken together is called the transcription initiation complex. Once everything is in place, the RNA polymerase can start transcription. All right. This is just showing you again. So this is the start site. So when we're talking about this, this would be minus 30, right? Remember we talked about the Tata boxes minus 30. You're generally minus 30 in eukaryotes, and then we have a minus 10 to minus 35 box. Usually it's minus 10 in prokaryotes. So TBP, Tata binding protein, finds and recognizes the Tata box. And then this TF means transcription factor. So there's lots of different transcription factors. We talk about those as general transcription factors that initiate transcription. So here's some other ones. They're very uniquely named. This one is D, this one is F, this one is H, this one is just 3. And like I said, there's lots of these things. I don't expect you to know all these, right? I'm just trying to show you how this occurs. So you should know the Tata binding protein. You should know that that recognizes the Tata box. I don't care that you know all these other initiation factors, right? Um, Also known as transcription factors. But I'm just going to kind of go through here. So we have the Tata binding protein recognizes that. The transcription factors bind to the Tata binding protein. That recruits transcription factor 2A, transcription factor 2B. That binds here. And again, I'm not going to test you on this. Once that occurs, then we have transcription factor 2F that's bound to the RNA polymerase, and remember it's 2, so that tells us it's eukaryotic because there's only one RNA polymerase in prokaryotes. And not only that, but it tells us what it's going to do. So in this case, remember polymerase 2, which you do need to know, is the one that makes mRNA. And I might ask you about polymerase 1 and polymerase 3 on the test as well. So make sure you know what they do, polymerase 1, 2, and 3. And this isn't a DNA polymerase, this is an RNA polymerase. Okay, so 2E, 2H, those are also recruited. All these things are in place, and that is initiation. And then we can have elongation, which comes next after initiation. So here's the elongation phase. The RNA polymerase separates from the GTFs. So they move, right? So here's the polymerase itself. This is general transcription factors is what that stands for, by the way. So all these ones that are shown here, besides the purple thing, this is the polymerase. All these are general transcription factors. F, the Tata binding protein, F, 3, F2F, 2H, and so on. So anyway, these are all they after initiation they're released, and the polymerase travels down the DNA, and it's just like I said, if it sees an A, it's going to put in a U. If it sees a G, it's going to put in a C, and so forth. Remember, it has to go in the 5' to 3' direction. All right. Polymerase 2 makes the primary transcript, or what we call pre-mRNA. So pre-mRNA is an mRNA. So we have our promoter, our coding region, and our terminator. So it's only the coding region that's going to be read into what we call pre-mRNA. This is in eukaryote, so it's still in the nucleus. It's going to get edited. Um, it's going to have caps put on it. It's going to have tails put on it. It's going to get cut up and then rearranged. So we call that splicing. And all that has to occur before it's allowed outside of the nucleus. So all this is occurring inside the nucleus. Okay, so when we say that these are co-transcriptional modifications because they occur while transcription is taking place. You may have learned that this occurs after transcription, but that's not true. So I'm here to tell you that it happens during. So so we know that this is the 5' end of the mRNA, right? Because transcription is in this direction. Otherwise, the promoter would be on this side. So this is the 3' end of DNA. 5' end of the mRNA, 3' end of the mRNA. So we have our 5', our 3' mRNA, pre-mRNA. On this 5' end, we're going to get a cap. It's going to be a G. It has a special structure, so most things are 2 to 5' linked. I'm sorry, 3 to 5' linked. This is 2 to 5' linked. And then we have a poly A tail. So here we're going to have lots of A's. That's what poly A means. Added to the end. And then splicing removes introns. So introns are regions. An intron is a region inside the RNA that's going to get cut out. So the intron would be cut out. Let's say that this is exon 1. This is intron 1. This is exon 2. This, in the end of this processing, it would only have the two exons, exon 1 and exon 2, linked together. The intron would get put into the lysosome and degraded to be recycled. And so we would have just the exon and intron linked together with our G cap, 5' cap, and our poly A tail. All right, so I'm just going to go over this. It's not that complicated. So here's what capping occurs. I'm not going to ask you to. There's enzymes that do this. They associate with a phosphate-rich region of the protein in the polymerase. And so once this occurs, this 5' cap is attached. And that's why it's called co-transcriptional, because it's done while it's being transcribed. Then while we're being transcribed, so the artist is showing you that these regions are the introns. Remember, introns get cut out. Exons stay. So these are the introns. They're going to get cut out through splicing. And then in the end, we're going to have that poly A tail added. So those are the 3 steps of modification. And so a lot of texts might say that this is post-transcriptional modification, but it's really co-transcriptional. And you can see that. Okay, let's talk about the capping. So on the 5' end of the RNA, so we have the 5' end and the 3' end, and remember that RNA is transcribed in this direction. So on the 5' end, we have a cap that's a G, which we talked about, and so when you're drawing out this things in the problem sets, make sure you add all this stuff in. And then, what's the purpose of the 5' cap? Well, once it leaves the nucleus, once the RNA leaves the nucleus, it goes to the cytoplasm C. There's a lot of nasty stuff out there. It's kind of like taking a voyage out of the harbor into the ocean. So lots of things could go wrong, and there's lots of nasty, like, whales and sharks and all kinds of stuff, and storms. So the cytoplasm isn't really that nice. It's not as nice as the house in the nucleus. And there's lots of enzymes that will degrade RNA. So remember, we call those exonucleases. And so they're gonna be able to cut up that RNA, and you don't want it cutting up your RNA, because if the RNA gets cut up, then you're not going to get any protein made, and that's the whole point. So we've got to get the RNA out of the nucleus and to the ribosome before it gets chopped up by all the nasty enzymes in the cytoplasm. And the G cap protects it from degradation by those enzymes. We call those exonucleases. So it also allows the mRNA to be exported from the nucleus. How do we know that? Well, if there's no cap on it, it won't leave the nucleus. So it's required to export it out through the nuclear pores from the nucleus to the cytoplasm. And then it helps the ribosome recognize the attachment site. So let's just pretend that this is, I'm going to use the word rat, just because I don't feel like drawing nucleotides. So if we read it this way, from left to right, when you look at that, you think of a rat, right? And that's a furry mammal, maybe with a tail, 4 legs, and so on. So you would want your ribosome reading it from left to right. You wouldn't want the ribosome reading it this direction, because that would be tar. And that is a completely different thing. You'd have a completely different protein that would have a completely different shape, that would have a completely different function. And you don't want that. So this is what the cap looks like. I'm not going to ask you to draw this or anything, but it's methylated. There's a reason for that. We're not going to go into too much detail of that. This is the nucleotide. Remember, this is RNA because it has 2 hydroxyl groups. And then this is numbered 1, 2, 3, 4, 5, just like we said. So on the 5' end, normally this is where the phosphate group is. And you can see this is a triphosphate, which is unusual because usually it's just a phosphate. And then this is linked on the 2', right, to 2', rather than 3' to 5', which is what we learned with DNA. So basically, it's just bending around the end of the RNA, kind of to make sure that it's protected. And that's, I don't care that you know this. I'm just telling you that this is the physical structure that's linked together to protect that cap. And that shape also signals it to be able to be exported out of the nucleus. All right, so the tail modification, there's variations in how many A's that can be added on here, and this is really simple. So if you have an exonuclease that's going to chop up your RNA no matter what do you want? What do you think is going to last longer? One with 10 A's or one with 6 or 5 A's? I don't even know how many that is. And the answer is this one, right? Because it takes a lot longer to chop up. It's an exonuclease, so it's cutting from the outside. This is the 3' end. It's going to take a lot longer to cut out 10 or 200 or whatever. So this is how you can vary your half-life of your RNA. The longer the RNA is out there, the more ribosomes. Again, ribosomes are like polymerases. You can have multiple ribosomes on the same RNA, reading it, converting it into protein. So you can make lots of protein from a single RNA, just like you can make lots of RNA from a single gene. So the more poly A tails, the more poly A's you have on your tail, the longer your half-life is going to be, the longer that's going to stick around. And the shorter is, so longer poly A tails increase your half-life. Shorter ones, if we shorten it, it decreases the half-life. And maybe you don't, maybe there's things that you want around a long time, like housekeeping genes, for example. You need the genes, the enzymes that are involved in glycolysis around all the time because that's how you make energy. So you would want those to have a very long half-life. But let's just say for fun, what if it's the gene, the protein that makes you shiver? So do you want that around all the time? When you get warm, you'd keep shivering if it had a really long half-life. So you want that to have a short half-life. And all of this has developed different half-lives through evolution. And not only that, but different environments. Okay, the polyadenylation of the tail. Again, I'm not going to ask you all this stuff. I'm just showing you this so that you know that these enzymes exist. This, I might ask you the sequence that it binds to. It's AAU, AAA. This is the pre-mRNA, so it's the 5 prime. This is the 3 prime end. I guess the polymerase is going this direction. So this is where the cleavage occurs for the cap. 5 prime end, and then 3 prime end. So this is. Oh, okay. So I see what's going on. So it's cleaving here. This is going to be junked. This is the 3 prime end that's going to be junked. This is where the cleavage occurs, and then the poly A tail is added on here. So that's what's going on here. And these are the enzymes that do it. I don't care that you know that. The enzyme is called polyadenylase. All right. Splicing. So we have introns and exons. The introns and exons, remember the exons are kept, the introns are not. So we just number this: exon 1, intron 1, exon 2, intron 2, exon 3. And so in the end, these introns are removed. That's done through a process called RNA splicing. We've already got our cap, we've already got our tail. So the last thing we have to do is splice. Now, the reason that you would do that is, like, you ever played with those magnets where that you can make sentences out of, so it has individual words and you can move them around on your refrigerator? Well, if you didn't have introns and exons, then you wouldn't be able to move, you wouldn't be able to shuffle these around. So if you have an exon 1 and an exon 2 and an exon 3, and let's say that this I don't know, the fat rat or something. I'm just making this up. So you could only use that one sentence if they were all permanently linked together, but you could switch it around. And we do that. So we can take exon 3 and link it to exon 2 and link that to exon 1. Again, it's going to be 5 prime to 3 prime. So this one is 3 prime or 5 prime, also 5 prime to 3 prime. So this would be very different. It's kind of like the rat and the tar analogy. You could also link exon 2 to exon 1 to exon 3, or exon 3 to exon 1 to exon 2. So there's all kinds of variations that you could do here to make, and the purpose of this is to make multiple RNAs from the same gene. All right, so the machinery that does this is called the spliceosome. It contains protein and the snRNAs that we talked about. Remember, RNAs can make those stem loop structures that we discussed. This is called a SMILE, nuclear ribonuclear particle, snRNP. And then there's other proteins that combine here. And basically, there's what we call intron-exon junctions. So there's a DNA sequence here that this recognizes, and there's a DNA sequence here that this recognizes. And the spliceosome basically brings it together. So when it brings it together, it performs a hydrolysis reaction, which cuts here, and then a dehydration reaction, which links the two exons together, and then this excised intron is sent to the lysosome for degradation. So that's how the spliceosome works. It's recognize these RNA particles are recognizing the DNA sequences at the intron. We call it the intron exon junction. And this is going to be really important because you could have mutations there that would cause it not to be able to splice. So here's the actual sequence. Uh, AGCU, AGGU, AGGU. They're both the same. There's an A in here that's recognized by part of the spliceosome. So right here. Oh, this doesn't show it. So anyway, we have these SMP U1 and U2. U1 binds here at the GU site. U2 binds here at the A site. This is within the intron. So this A site is within the intron. Then these are get other proteins bind to cause the RNA to, or the RNA transcript to bend. And when it bends, it brings these together so that you can have the hydrolysis and dehydration reaction. So we have the reaction here and here. It's a complicated chemical. You can see. I'll show it to you. And then this is what this reacts here, and this is what causes that lariat structure. And these are the spliced exons. This isn't a chemistry class, so I'm not really gonna make you responsible for this. But you can see the hydroxyl attacks at this phosphate and causes the reaction to occur. That cleaves this off, leaving this as a negative charged oxygen, and then this oxygen actually binds here, so we have that. Then this oxygen reacts here with this phosphate at this point. So we have the phosphate linked in between these two, and that's part of the phosphodiester backbone of the RNA. Okay, so this is just showing you an example of alternative splicing. In this case, we have alternative splicing in a rat tropomyosin gene. So remember, myosin has something to do with muscle. And so we have striated muscle, smooth muscle, brain, tissues, and so on. And so these are all from the same gene, right? This is the gene, this is the sequence, and if we couldn't do alternative splicing, this is all you can make out of it. But we can. So we can link this to this to this and this and this. And then we make a different transcript. And that's going to be turned into a protein, right? And that protein is going to be different than this protein and so on. So this is an example of how we can make 5 different proteins from a single gene by alternative splicing. All right, so this is the intron exon junctions we talked about. And so the spliceosome recognizes all the splice junctions, and it recognizes each of these and can cleave them out. So you can have this one cleave the whole thing out. You can have this one cleave here, and so on and so forth. You can have a splicing repressor that binds here and causes it not to recognize this intron exon junction. And in that case, it's going to splice from here to here because this is the next junction, right, to here. And so the splicing machinery will cleave out both. In this case, this would be intron 1 and exon 2 and intron 2 as well. So all 3 of these would get cut out, and then you just have 1, 3, and 4 put together. All right. So exons can put things in different orders. We talked about that, and generally, when we talk about this, we have There you go. We talk about domains. So here's an example. We're going to talk about this later on, but let's say that we have something called the estrogen binding receptor. That has three domains, right? So it has one domain that binds estrogen. I don't know why this pad's being so weird. Binds estrogen. It has another domain that is a DNA binding domain. And then it has another domain that is a transcription factor. So it's going to go into the nucleus, find a DNA site that recognizes here it's a DNA binding domain, and it's going to bind it within a promoter. That promoter is going to be within a gene that responds to estrogen, and then it's going to bring along with it the transcription factor needed to initiate transcription, to cause the RNA polymerase to transcribe that and then make that RNA, which is going to be exported out of the nucleus and then read into a protein as a response to estrogen, right? And the same thing for all other hormones. So when we talk about these exons, we also generally consider them being domains as well. And in this case, it's the same. All right. So when we have the mRNA, uh, the mRNA is longer. I don't know why it's doing this. mRNA is longer. It's trying to draw at an angle. I apologize. So this mRNA is the coding region. Remember, we talked about the promoter and the coding region and the terminator. So this is the coding region right here. So that's what this is representing. But within the coding region, not all of this is going to be turned into protein. And we'll talk about this when we get to translation. So there is untranslated areas on the 5' end. It's called a leader. And then there's an untranslated area on the 3' end. These ends are important to regulate where the mRNA goes. So remember, I don't know if you remember this from intro biology, but there are two kinds of ribosomes. There are free ribosomes that are just sitting around in the nucleus, and generally those free ribosomes code for things that are going to stay in the nucleus. And then we have ribosomes that are bound, and those ribosomes code for things that are going to be exported. And they're bound to the endoplasmic reticulum. This should be a review. So what tells the mRNA that's coming out of the nucleus? So here's the nucleus. Here's the endoplasmic reticulum. It's rough because it has ribosomes attached to it. And then we have free ribosomes hanging out here. So what tells the mRNA to either go to the endoplasmic reticulum because it's going to be exported out of the cell, or go to the free ribosome because it's going to stay in the cell. And it's the sequences in here that direct where that mRNA is supposed to go, and also how it's supposed to be modified, because we can have other modifications that occur too. So only portion of this is actually going to be made into a protein. And we're going to talk about, so this is the start site of, not of transcription, but of translation. And then there's a stop of not transcription, but of translation. So a lot of students when they start learning this, they get things confused. So remember that there's like 3 piles here. You have a pile of things for replication. You have a pile of things for transcription. And then when we get to the next chapter, you're going to have a pile of things for translation. Okay, so there's other things that we're going to talk about. RNA interference. So that is when you have an RNA that's made. That RNA can interfere because it's complementary to the messenger RNA. So this is a small interfering RNA, miRNA, and that codes to complement part of the mRNA, and that targets it to get destroyed. So this was discovered by a guy named Rich Jorgensen, and he actually worked at the University of Arizona. He was studying petunias, and he was trying to insert a gene to make it more purple. What he was trying to do ultimately is make a blue rose. He wanted to show his investors that he could make a rose that was blue. At, at the time, and I think it's still true, blue roses don't exist. So they were able to make roses blue, but it wasn't just a flower. It was the flower and the stem and the thorns and everything. And that's not really what rose enthusiasts are after. And it's kind of a big deal. Like it's super rare to find a blue rose. It's worth millions of dollars. So he was like, I'm gonna make a blue rose. When to do that, he was using petunia genetics, which are a lot simpler than rose genetics. And so he knew that certain genes would be controlled for flower color, like Mendel. And so what he decided to do is just put extra copies of that gene in to make it more purple, or darker copies of that gene in to make it more purple. And when he did that, he put it in backwards. So imagine that if you had a gene that was this, right? And this is the um, this is the coding strand. So this is the non-coding strand. So it would be read in this direction, right? But if you flip this gene around, so let's say if you rotated this gene, then the promoter would be over here and it would be read backwards. And so because of that, you would make a complementary RNA to that mRNA. And that's what he did. So when you get double-stranded RNA, it signals the cell that that's probably a virus, and it turns on a whole system to destroy that RNA that it perceives as a virus. So this is how it works. There's a enzyme called, it's a complex of enzymes called dicer. It binds to double-stranded RNA. It chops it up into siRNA, which is small interfering RNA. Let's go. Oh, I don't know what just happened. My computer froze up. So anyway, we have the small interfering RNA that is made into single strands that bind to a RISC complex. That RISC complex binds to the target mRNA, and that degrades the target mRNA. So it's kind of cool. They've used this to treat diseases like macular degeneration and stuff like that. It's relatively new, um, but basically what you do is you just make a complementary RNA or a double-stranded RNA, inject it into a cell. The cell freaks out, thinks it's a virus, and then it destroys all complementary copies of that RNA. And it doesn't matter if it's virus or not. Anything that looks like it, it's going to destroy. And so when Jorgensen did this in his petunia, and he wanted to make it more purple, he flipped a gene backwards, made uh, the regular gene and the extra gene complement, the mRNAs complement one another. That made double-stranded RNA, which told the cell it was a virus, and so it destroyed all copies of purple, regular copies, any whatever. And so by default, in petunias, because they're simple genetics, the flowers were white. So that's the RNA interference. And that's the end of this lecture. So that covers all the RNA stuff and all the transcription stuff. We're going to pick this up. I'll record another lecture probably this weekend. And that's going to talk about proteins and then translation. So converting this RNA in the cytoplasm C via ribosomes into essentially flesh and blood.